Hey guys, welcome back. Today I'll be discussing about the topic the reaction catalyzed by nitrogenase during biological nitrogen fixation. Today I'll be focusing on these subtopics: biological nitrogen fixation, development of root nodules, and then key aspects of nitrogenase enzyme and oxygen scavenger. After that, some important questions for NEET, which are from previous year question papers. So let's begin the session. Here, first of all, what is biological nitrogen fixation? It's very simple. Reduction of nitrogen to ammonia by living organisms is called biological nitrogen fixation. Here you can see this is nitrogen and here it is reduced to ammonia, and this reaction is catalyzed by nitrogenase enzyme. And one of the very important aspect here is only certain prokaryotic species, only certain prokaryotes contain this nitrogenase enzyme in them. Okay, and this nitrogenase enzyme is capable of reduction of nitrogen into ammonia, and this is exclusively found in prokaryotes, only certain prokaryotes. Okay, so this is what we call biological nitrogen fixation. Now moving to the next, the nitrogen fixing microbes could be free living as well as symbiotic. Okay, let's discuss about the examples. Free living bacteria, examples are Azotobacter, Bergerichia and Rhodospirillum. So these are the free living nitrogen fixing bacteria. Now here free living cyanobacteria are Anabina and Nostoc. Okay, so these are examples of cyanobacteria. Okay, apart from these free living microorganisms, okay, symbiotic bacteria like Rhizobium and Frankia are also playing important role in this biological nitrogen fixation. And here you can see all these examples which are mentioned here are prokaryotes. Okay, and now here several types of symbiotic biological nitrogen fixing associations are known, but the most common symbiotic association on roots, okay, is as nodules. Okay, so this is the most common association that we find, okay, between the bacteria and the root nodules, okay, roots of the leguminous plants and some of the non-leguminous plants. Here Rhizobium bacteria has association with the roots of leguminous plants. There are several examples: alfalfa, okay, sweet pea, okay, lentils, garden pea, okay, broad beans, clover beans. All these are examples of leguminous plants. Okay, so Rhizobium bacteria has association with leguminous plants, and Frankia has association with a non-leguminous plant called Alnus, important for exams. Okay, Frankia, it is a symbiotic bacteria, has association with root nodules, roots of non-leguminous plants, and the example is Alnus. Okay, so this is about the nitrogen fixing microorganisms. Okay, now moving to the next, Now let's discuss about the development of root nodules. Okay, root nodules are near spherical outgrowths on the roots. Okay, so root nodules are the near spherical outgrowths on the roots of the plants. Okay, and these root nodule formation involves a sequence of multiple interactions. Okay, so this is what you should understand. Okay, so root nodule formation involves a sequence of multiple interactions, okay, between rhizobium and the roots of the leguminous plant, host plant. Okay, now rhizobia multiply and colonize the surroundings of the roots. You can see here, this is a root epidermis, these are the root hairs. Okay, rhizobium bacteria multiply and colonize. You can see here the colonies of bacteria are seen here. Okay, now they get attached to the epidermal and root hair cells. These rhizobium bacteria, okay, they get attached to the epidermal cells and the root hair cells. Okay, after that the root hairs curl and the bacteria invade the root hair. You can see this. Okay, the root hair is curling like this. Okay, so after this curling of the root hair, okay, the bacteria invade the root hair. Okay, so this is how the bacteria is entering into the roots. Okay, now after this, moving to the next, an infection thread, important feature. Okay, an infection thread is produced carrying the bacteria into the cortex of the root. You can see here inside this root hair the infection thread. This is how it is seen. Okay, so this is the infection thread. An infection thread is produced carrying the bacteria into cortex of the root. Okay, now the bacteria are released from the thread into the inner cortex and pericycle cells in the roots. Okay, after that the differentiation process, the differentiation of specialized nitrogen fixing cells occur, and because of this differentiation process, okay, the root nodule is formed. This is the mature root nodule. Okay, the nodule thus formed establishes a direct vascular connection, okay, with the host. Okay, important feature. The root nodule establishes direct vascular connection with the host for exchange of nutrients. Okay, so this connection is for exchange of nutrients. Okay, now the nodule contains all the necessary biochemical components such as, okay, here nitrogenase enzyme and leg hemoglobin. Okay, leg hemoglobin. Okay, so these are very much essential for the biological nitrogen fixation. I repeat this statement again. The nodule, this mature root nodule, contains all the necessary biochemical components such as nitrogenase enzyme and leg hemoglobin, which is an oxygen scavenger, about which we will discuss in the coming slides. Okay, now moving to the next, nitrogenase enzyme. Let's discuss few important aspects about this enzyme. It is a molybdenum iron protein. Important for exams. Okay? It is a molybdenum iron protein. And this enzyme, the nitrogenase, catalyzes conversion of atmospheric nitrogen to ammonia. You can see here the chemical reaction. This is atmospheric nitrogen. It is converted into ammonia, and this reaction is catalyzed by this nitrogenase enzyme. Okay? And one of the very important aspect is ammonia synthesis by nitrogenase enzyme requires very high input of energy. Okay? Very high input of energy. Okay? Here you can see for the formation of two ammonia molecules, 16 ATPs are required. It means for the formation of one ammonia, how many ATPs are required? The answer is 8 ATPs. Okay? So 8 ATPs are required for each NH3 formation. Okay? So now this energy required is obtained from the respiration process taking place in the host cells. Okay? So the respiration in host cells. Okay? Because of this respiration in host cells, energy is released, and from that ATP molecules are utilized for this biological nitrogen fixation. Okay? Now moving to the next. Here you can see how the reaction is taking place. This is enzyme nitrogenase. Okay? Now here is the substrate nitrogen. Okay? Enzyme is nitrogenase and the substrate is nitrogen. This is an enzyme substrate complex. Okay? Binding of substrate to enzyme results in the formation of enzyme substrate complex. Okay? Now you can see addition of hydrogen. Addition of hydrogen. These are nothing but the reduction reaction. And because of reduction reactions, this atmospheric nitrogen is converted into ammonia. Okay? So here after the formation of ammonia, ammonia is released, and again the enzyme, the free nitrogen ase enzyme is again available for next reaction of biological nitrogen fixation. So this is how the reaction is catalyzed by nitrogenase enzyme. Now moving to the next. Here oxygen scavenger, one of the very important aspect in biological nitrogen fixation. Okay? If you cut root nodules, okay, at the central portion, you will find, okay, red or pink colored central portion. Okay? This is because of the presence of, okay, oxygen scavenger, that is leg hemoglobin. Okay? That is leg hemoglobin or leguminous hemoglobin is present in root nodules. Okay? The nodules have adaptations that ensure the enzyme is protected from oxygen. Okay? Because this enzyme is sensitive to molecular oxygen. The nitrogenase enzyme is sensitive to molecular oxygen. So the leg hemoglobin, the oxygen scavenger, is present inside the root nodules. Okay? So the nodules have adaptations that ensure the enzyme is protected from oxygen, and this oxygen scavenger, the leg hemoglobin, is playing important role in this. To protect the enzyme the nodule contains, okay, this oxygen scavenger. Okay, so this is all about today's topic. Based on the topics that we have discussed till now, let's discuss some important questions that are previously asked in NEET exams. Okay, so let's discuss some of the important questions for NEET exam. Okay, question 1. The products of reaction catalyzed by nitrogenase in root nodules of leguminous plants is or the products. Okay, so this question was asked in NEET 2020. So here option 1, ammonia alone. This is wrong. Okay, you can see here. What do you find here in this chemical reaction? Do you find ammonia alone? Do you find ammonia alone? No, you will also find hydrogen. Okay, so the answer for this question, okay, it is not ammonia alone. This option is wrong. Second, nitrate alone. No, nitrate is not formed in this reaction. Ammonia and oxygen. No, oxygen is not formed here. Ammonia and hydrogen is the right answer. So the more appropriate option for this is fourth option. So the products of reaction catalyzed by nitrogenase in root nodules of leguminous plants are ammonia and hydrogen. Okay, now moving to the next question after this. Select the mismatch. This question was asked in NEET 2017. Okay, here option 1, Frankia. It is a bacteria, nitrogen fixing bacteria. Okay, Alnus. Yes, it is a symbiotic bacteria, forms root nodules in non-leguminous plant Alnus. This is correct. So we should find out mismatch. This is actually the correct match. Okay. Next, Rhodospirillum and mycorrhiza. Rhodospirillum is a nitrogen fixing bacteria, but mycorrhiza is not associated with this. Okay, mycorrhiza is a symbiotic association, okay, between algae and fungi. Okay, so this is wrong. This is wrong. So the most appropriate option here will be second option. Okay, this is mismatch, not correct. Okay, if we check the other options, Anabina, nitrogen fixer. Yes, it is a cyanobacteria. It is nitrogen fixing cyanobacteria. Okay, if we check the other option, Rhizobium, alpha alpha. Alpha alpha is a leguminous plant, and Rhizobium has association with leguminous plants. So this is also correct. So the mismatch here is second option. Okay, now moving to the next question. The first stable product of fixation of atmospheric nitrogen in leguminous plants. The first stable product here. This question was asked in NEET 2013. Okay, option 1, NO2 minus. This is wrong. Nitrite is not the first stable product. Okay, ammonia. Yes, ammonia is the first stable product. This is correct. If we check the other options, nitrate. No, this is not the first stable product. Glutamate. No, this is also wrong. So the more appropriate option is option 2. That is the first stable product of fixation of atmospheric nitrogen in leguminous plants is ammonia. Okay, now moving to the next question. Question 4. The function of leg hemoglobin in the root nodule of legumes is here. Option 1, expression of NIF gene. This is wrong. What is NIF gene? It's very simple. NIF gene. Okay, this is nothing but, okay, the genes encoding enzymes. Okay, genes encoding enzymes, okay, involved in, okay, fixation of atmospheric nitrogen. Okay, so those genes are called NIF gene. So expression of NIF gene, this is wrong. Okay, the function of leg hemoglobin in the root nodules of legumes is not the expression of NIF gene. Okay, second option, inhibition of oxygenase activity, no, this is wrong. And the third option, oxygen removal, oxygen removal is the right option. Okay, in the absence of oxygen, okay, nitrogenase enzyme can catalyze the biological nitrogen fixation. So this is the right option. Okay, if we check the fourth option, nodule differentiation, no, that NIF gene do not have role in nodule differentiation. The most appropriate option here is the third option. Okay, the function of leg hemoglobin in the root nodules of legumes is oxygen removal. Okay, this is all about today's session. If you are new to my channel, subscribe for more live sessions. I'll be back with another session. Have a great day.